people will join as we start. So we're going to start today. Our webinar today is on wastewater surveillance and humanitarian WASH. And we'll have 4 speakers. We'll have Marlene Wolf from Emory University, Ross Tomlinson from the State of Palestine WASH Cluster, Yapi Marius from Afrahoon, and Rochelle Holm from the University of Louisville in Malawi. Captions are available in English and French and Spanish, so feel free to go to captions to select language if you would like. This webinar is part of the Tufts Research to Action project, where we're helping with leading research to action with this resource center on the Global WASH Cluster website, a research help desk, helping anything from helping design research to analyzing data, a webinar series, which is where we are today, and also training on research in LMIC context. And we're thrilled to have speakers today with us on current wastewater surveillance. So I will quickly move through. I'm going to stop my share and I'm going to invite Dr. Marlene Wolf, Professor Marlene Wolf from Emory University to do the first presentation. Thank you.
Hello, everyone. Good morning, good afternoon, good evening, depending on where you are. I'm going to share my screen and hopefully you can see it. I don't know why. Okay, there we go. Okay, so I'm going to talk a little bit about an overall context for wastewater monitoring and some of the work done in high-income countries from my perspective, doing a lot of wastewater monitoring in the United States and some of the differences. So first, I just want to set the stage for what this work is that we're doing. Wastewater can provide insights on infectious diseases because when a person has an infectious disease, they shed the pathogen in bodily secretions quite commonly, right? And so then in waste, however it is collected, we have a composite biological sample that can provide information about the whole community just by sampling from that one location. That provides community insights when clinical data might be slow, incomplete, or unavailable, which is very frequently true all over the world. The amount of genetic material in wastewater from these infectious agents is very often related to the number of people who are infected for a lot of different diseases, is what we found. So this can be a really broadly useful tool. The way the testing process typically goes is that wastewater and environmental samples are tested by targeting the genetic material of the pathogens of interest. So the general flow of the process is that samples are collected. This is an aerial view of settling tanks at a utility in the United States. Those samples are then prepared, so they are purified and concentrated, and then the DNA and RNA is extracted. Then there's detection and quantification that happens through molecular methods like PCR. And then sequencing and characterization of viruses can also be done from these samples. And then finally, data analysis and sharing. And what you're seeing here, the blue, is the quantitative amount of copies of SARS-CoV-2 RNA found in a wastewater sample. So you can see how it basically gives us the shape of an epidemic curve. In a lot of cases, the same process can be used to collect and concentrate a sample that can be tested for a lot of different targets, which is really useful and cost-effective. The basic history of wastewater monitoring, I think, is important to look to because prior to the COVID pandemic, wastewater monitoring was focused on low and middle income countries and on enteric diseases like polio. This started in the 1940s and has long been done mostly through culture-based methods. So quite different than a lot of what we're talking about when we talk about this sort of new renaissance of wastewater monitoring. That monitoring was not focused on high-income countries and in fact has barriers in countries where polio has been eradicated. If we detect polio in wastewater in the US, a whole chain of things starts to happen that's very bad. There's a lot of containment process. And so we really actually can't very effectively do polio monitoring through wastewater in the United States. So this really, is not coming from a place of a history really in high-income countries for the most part. During COVID, the high rates of shedding in feces of SARS-CoV-2 made people look to the sewers and think, well, this could be a useful tool for this case too. Advanced molecular techniques have also improved sensitivity recently, and so wastewater surveillance, therefore, has been adopted in many high-income countries. And now we're looking beyond COVID to the expansion of monitoring of sewage worldwide for lots of other pathogens as well. Many pathogens are shed into wastewater. So again, I just want to say there are so many different things that we can look for because we're talking about feces, urine, sputum, vomit, blood, skin, mucus. Everything really appears in wastewater. And in network sewer systems, many drains lead to the sewer. So there are a lot of different things we can be creative about monitoring and use as a complement to existing modes of surveillance. For network sewer systems, we generally have well-defined system inputs, some consistency in flow, even if not naturally, it is managed to create consistency. And it's usually separated from the environment. Some systems accept environmental inputs and storm overflow, but most in the United States do not. And so that means that quantitative measurements are consistently useful. We can compare day to day and week to week the concentration we observe, and that is related to the number of people who are sick in the community. If you don't have a sewer, that quantitative relationship is a little bit more difficult. But this environmental surveillance is a broader term that includes monitoring outside of the designed and networked sewer systems. No system is perfect. And so this can be a little trickier for communities that are on septic, for example, where it's not a very large group of people who are contributing to that same sample. But places with informal systems, there is a lot of sampling, and this has been done, again, for a long time. So now we're looking at sort of learning how to take some of the new tools that we've developed and have this learning go back and forth between high-income countries and low and middle-income country settings to create kind of a global network. These are just a couple examples of places where we've sampled at open drains in Bangladesh, latrines at schools in northern Ghana. I want to quickly show you these plots of SARS-CoV-2. So I showed this one before. The blue is the concentration of SARS-CoV-2 viral RNA in wastewater, and the red is the 7-day average COVID cases per 100,000 people. And so what you can see here, this is another color blue, but it's the same thing where we're seeing the blue is the concentrations in wastewater and the red is cases. The time scale on these is different, but this is the same time period here and here where that red line representing cases seems to like disengage a little bit with the wastewater data. And what we're seeing, what happened there is that there was a change in testing practices in the United States that drastically altered the accessibility of tests and the way that they were counted. And so what we can see in the decoupling of the wastewater data from the clinical data in these two examples is that the wastewater data is continuing to give us a consistent picture of the number of infections in the community when those testing practices change. So it's very robust to some of these changes that can be really challenging, especially in, in outbreak settings. I just want to share that there are so many other things that we're expanding to now, and I'm not going to go through each of these plots in detail. I'm happy to share these slides. But these are examples of work that we've done on influenza, RSV, and Mpox, demonstrating the relationship between wastewater data, which is on the top in all of these plots, and case data, which is on the bottom. And so we've been able to demonstrate that this is really effective for monitoring all these different diseases and put that into practice. So for program that I'm running right now in the United States, I realize it's November. I'm sorry, that says September. This is still true. We are monitoring for all of these different diseases in wastewater across the United States. And we've done research on a lot of other targets. Just a few of those are HIV, dengue, measles, and we're continuing to expand to other things that can be monitored in wastewater and that are of global relevance for us to be tracking outbreaks. So as some quick takeaways, wastewater can provide rapid, valuable data on occurrence and outbreak characteristics at a community level for many diseases. It can be quick because we are looking at that one sample for the whole community. Public health and academic practitioner partnerships provide opportunities to quickly respond to new concerns by implementing wastewater testing. So as an academic, being in conversation and collaboration with practitioners is incredibly important for us to be able to turn this work around and do it quickly. And a feedback loop between tools in use in high-income countries and low and middle-income countries can ensure a global system that is prepared to address existing and emerging threats with the learning coming in both directions. Those partnerships are absolutely critical to developing this field further. And I will stop there. Thank you.
I wanted to say thank you so much to Marlene for providing such a good overview of wastewater surveillance within a high-income context setting and what the possibilities are. We're going to hand this over now to the next three speakers, the first of whom is Ross Tomlinson, to speak about implementing wastewater surveillance within humanitarian contexts and low-income contexts. So Ross, thank you.
Thanks, Daniele, and hello, everybody. So my name's Ross Tomlinson. Can you hear me? I'm the National Cluster Coordinator for the State of Palestine, and I'm sure you're all up to speed with what's going on in Palestine, particularly in Gaza at the moment. Thanks. Unfortunately, I don't have a slideshow for you, so you're going to have to hear my words and picture what I'm talking about. So pre-conflict in Gaza, you have a territory, say, the size of Manhattan, 20 kilometers by 50 kilometers long, with about 2.2 million people, highly urbanized. About 80% of households were connected to the sewage mains, and the other 20% had maybe a septic tank or some kind of on-site sullage system inside them. And these households were connected. The sewage system had a lot of a series of pumps connected up to the 6 wastewater treatment plants spread across the 5 governorates of Gaza. In the north of the Wadi, you had 2 plants, and south of the Wadi, you had another 4 plants. As a result of the conflict, we lost all electricity supply, and that meant that the high consumption wastewater treatment plants were all switched off. They were also put in the less desirable locations of Gaza, which tended to be on the border with Israel, and so have become located in what we can call a danger zone. You can't go there. They are prohibited sites. Anyway, the volume of fuel that they'd used to operate would be too much compared to what we've got available. So no wastewater treatment has been taking place in Gaza since the start of the conflict. A little bit in Rafah during certain months when 1.5 million people were crammed into the southern governorate of Gaza. They were using that treatment plant for a short time, but pretty much most of it is being pumped out into the sea at the moment. So what was the priority in terms of wastewater sampling? We came under severe pressure or extreme pressure to actually start doing the testing. I believe that the testing had happened systematically before the war, but had stopped about 6 months beforehand. And so UNICEF conducted needed to arrange the approval to do the testing to start off with. We had to navigate security approvals with the Israelis in order to go and do the testing. And particularly important in this case was to be able to send the samples out of Gaza to an appropriate laboratory. Gaza is effectively a highly controlled environment. Nothing happens without Israel okaying it in terms of what materials get in terms of the movement of people around. So there was quite a long process of actually getting the right approvals in order to do the testing. When we could, we were able to test in the southern 3 governorates, where the bulk of the population are living in makeshift camps or in designated shelters. So this is Khan Yunis and Deir al-Balah. They took 8 samples in total, and I'll tell you the results in a moment. The 500,000 people who were still living in North Gaza, we were unable to do any testing there. So the results. Of the 8 samples, 6 came back positive for polio, polio being endemic in the area, but having had no cases in Gaza for many years. And so this resulted in, first of all, the opportunity to actually bring the humanitarian community together in order to run a polio vaccination campaign. Therein, you go into the same process of approvals across all of the authority mechanisms, the challenge again to get the polio vaccine in, to get the vitamin A, which we did as a supplement at the same time, and then to be able to negotiate the space within Gaza in order to do the vaccination. And pretty uniquely, in terms of this particular response, we were able to get clearance for a set geographic zone, and within that zone, polio vaccination was going on. Teams were able to go out and speak to the population, loud hailers. It was promoted in advance, obviously, and children under 10 were brought in for the vaccination. And then we were able to move that to a second location, and we were also able to operate that in the north. And if my numbers are correct, in phase 1, we were trying to achieve about 90,000 under 10s, and we were able to succeed with that. Phase 2, a month later, was basically a repetition of the same thing. And we were able to do south of Wadi Gaza without too many problems. North of Wadi Gaza, we had a major military offensive happening in the northernmost part. We had a restricted zone for running the vaccination campaign, but we're still able to get up to 85, and I think because I left at that time, we're able to get the full amount in. So what were the challenges? Mainly the political approval in order to be able to run such a vaccination campaign. In terms of doing the sampling, no real restrictions except for negotiating the samples out in order to do the testing. In fact, the powers that be were quite keen for us to have polio results come in. And what else can I say? I think from an operational perspective, the big lesson is that being able to do wastewater treatment should be key and put straight onto the agenda. How do we promote it as a key task? And the other was that it's actually an amazing opportunity for multi-agency cooperation, and it opened doors for us. Over.
Yeah, I wanted to say thank you, Ross, for that description of a program that the testing led straight to an intervention. And thank you so much for describing that. So we've heard from Dr. Marlene Wolf about kind of the general overview of wastewater surveillance in high-income countries and a bit of low-income countries. And then Ross on talking about a specific example in Gaza of using wastewater surveillance for practice. And now we'll move into Dr. Yapi Marius from Cote d'Ivoire, presenting on a project testing in for SARS-CoV-2 and HPAI in Cote d'Ivoire. Thank you, Dr. Yapi.
Good morning, everyone. Thank you. My name is Yapi Maris. I'm in Cote d'Ivoire, and I work in the National Public Health Institute. And actually, we work with the university. We work on the project we named Tufts University. So I will present to you this activity of stop spillover about the wastewater. So the title is the SARS-CoV-2 and HPI waste effluent surveillance in Abidjan. What is stop spillover? Stop spillover working with country stakeholders to understand and address the threat posed by the zoonotic viral diseases and reduce the risk of viral spillover and spread. What is the step? We have three: strengthen capacity. Stop spillover will strengthen capacity in priority countries to first, monitor, analyze, and characterize the risk of priority zoonotic virus spilling over from animal to people. Secondly, develop, test, and implement intervention and policies to reduce the risk of priority viral zoonotic spillover. And third, if spillover occur, mitigate the amplification and spread of priority zoonotic viral diseases. By definition, spillover risk is not highest in area with sewage system. So, concept of wastewater sampling for surveillance in spillover is novel, very novel. What is the process for waste effluent sampling? The first step is to review the research. And after that, we identify likely place of highest virus prevalence in the environment. In third position, we identify media to be tested. And after that, we identify exact testing location. We determine sampling method. And finally, we safely collect samples and transport them to the laboratory to ensure that we use appropriate testing for sample media and method. So who will test and what to test where? In Côte d'Ivoire, we identify 3 locations to test the wastewater sample. The first location is an open small canal that contains human wastewater in Abidjan. And in this location, we test COVID-19 with Institute Pasteur Lab. And to test this, we use 2 methods, the active and the passive method. The second location is an open, very small waste streaming that contains chicken slaughtering, liquid waste in market in Grand Bassam. Grand Bassam is an area which is near the big Abidjan. And at this location, we test avian influenza SPI. And we test this with the Lanada lab. Lanada is the animal lab in Côte d'Ivoire. We use the passive method. And in the third location, we have the main wastewater exit pipe on district hospital in Man. Man is another area which is far from Abidjan, and we identify the wastewater of the hospital, and at this location, we have to test the Lassa virus with the passive method with Institut Pasteur lab. So in this photo, you see the canal that I talked about. We have the wastewater in this canal. Secondly, we show how we put the bucket in the canal to do the grab sample. Here, we show the cotton and the stone in the wastewater. We tie the rope with a stone and we put it in the wastewater. We put the cotton in the wastewater. Here you show, this photo will show you how we tie the rope with the stone. About Lanada testing about avian influenza, this is only the passive method, and we show you how we put the cotton in the wastewater with the rope. The second photo is again the same thing. So what is the result about this method, these two methods? About SARS-CoV-2, we have positive sample about passive sample method. The positive sample is week 2 to week 5 and week 7 to week 8. But the active sample, for the active sample, we have positive sample with week 1, week 3 to week 9, and week 11. So the total result is 13 on 24 active sample positive and 9 on 24 passive sample positive. About avian influenza, the result is week 2 positive sample and week 10 positive sample. So about avian influenza, we have 3 positive sample on 24 sample. What is the success and the challenge we have with this activity? The successes is that we have a large international collaboration, a proof of concept, and a low cost and highly effective. We have to continue to develop development to democratize access to wastewater sampling. And about the challenge, laboratory supplies and PPE were imported in Côte d'Ivoire. So we have more training and supplies needed for quantitative PCR research, and we have again challenge about sequencing. To summarize, what we want to say is there is a need to extend the benefit of wastewater surveillance to areas without safely managed sanitation. We developed methods to test waste effluent stream in Côte d'Ivoire and successfully used this method to identify human and non-human animal-derived pathogens. Waste effluent surveillance is a promising technology for expanding the benefit of wastewater surveillance to health and humanitarian context. Thank you very much to hear me. So I wait for your question to give you again over response. Thank you very much. Over.
I wanted to say thank you so much, Dr. Yapi, for sharing that work in Côte d'Ivoire. I very much appreciate that example, along with the example that Ross gave. There are a number of questions in the Q&A. We have one more presentation of another example from Malawi, and then people will be writing answers to the questions in the Q&A, and then we'll have time to bring some of them back in. So for our last example, I'd like to turn it over to Dr. Rochelle Holm. And Rochelle, thank you.
Good afternoon from Malawi. My name is Rochelle Holm. I'm an environmental scientist with the University of Louisville, but I have a background in water, sanitation, and hygiene, and the intersection of these for human health. So I've lived in Malawi for more than a decade and played with a lot of pit latrine sludge. So I'd like to share a few lessons learned with you. Prior to the pandemic, I would ship samples of pit latrine sludge out of Malawi under a material transfer agreement for pathogen analysis. This was costly and results took months to receive back. Research questions in this earlier work centered around such as the pit latrine layers and pathogen detection. Essentially, can I scoop from the top of a pit latrine? Yes, even those maggots in this photo, or do I need to pump from the bottom sludge to get a representative sample? Our analysis showed that we could just scoop from the top, which makes sampling logistically much easier for pit latrines. A household pit latrine is like a time capsule of the health of the users over the last 3 to 5 years. This also matters for healthcare delivery in these areas of Malawi, where there is only one large government hospital and drug supply is managed at a national level by the government, despite that there may be regional differences in pathogen detection across the country. In 2022, the University of Louisville partnered with the Malawi University of Science and Technology to set up multi-pathogen baseline testing. In 2024, we collected samples weekly at about 10 sites. 7 of these are at the Zaleka refugee camp in the central region, and 3 of the sites are near the Malawi University of Science and Technology campus, where our primary wastewater lab is located. Most of these sites are pit latrines or septic tanks, though one hospital does release to a municipal system. Mostly though, due to limited supplies, we are running culture methods for cholera weekly. We do run pathogens on PCR only periodically, roughly on a quarterly basis. So we're really focused on cholera due to the recent cholera outbreak. Though a few months ago, the current cholera outbreak was declared to have ended, we still continue to monitor cholera on a weekly basis. Even in just these last few weeks, new cholera cases have cropped up, and we'll soon be starting rainy season, so we anticipate the wastewater detections again soon. In 2022, the WHO did confirm a wild polio virus type 1 case in Malawi, but we don't include that in our WBE work because we are not part of the global polio laboratory network and we cannot obtain PCR primers and probes. So we focus on what we can do with wastewater surveillance, such as cholera. Malawi hosts more than 50,000 refugees and asylum seekers, with a monthly average of 300 new arrivals. Almost half of these are children. The camp was initially established to host between 10,000 and 12,000 people. So currently I am an advisor to the UNHCR to examine the potential applications for using wastewater and environmental surveillance to monitor infectious disease agents in the camp. We focus a lot at the camp on what we can do with prevention monitoring, or where a patient might respond to treatment. The camp was broken up into several zones for sampling. Both pit latrines and a vacuum tanker were sampled. Community sensitization was conducted prior to sampling. Samples were analyzed at the Ministry of Health, which was set up for analysis specifically for this project. But about 10% of the samples were still sent to the University of Louisville and the Malawi University of Science and Technology lab for quality assurance. Data was reviewed every Friday with stakeholders, and in the event of positive results, these stakeholders were ready to work together to target necessary interventions like vaccinations, hygiene promotion, and bucket chlorination. However, data was under a strict non-disclosure agreement to ensure that there was no stigma to camp residents or that the data was not leaked to the media. Here's a recent video where we are conducting pit latrine sampling for pathogens to give you an idea of the sampling method. Funded by the NSF, we just finished data collection for Malawi wastewater-based epidemiology public opinion research, inclusive of national and camp respondents. Our findings suggest the Malawian and Malawi camp residents support ongoing wastewater and environmental surveillance for public health with few concerns. Results indicate high public support for surveilling communicable diseases, deadly diseases, environmental toxins, healthy eating, illegal drugs, mental illnesses, and prescription drugs. There is some opposition for surveillance at businesses and religious organizations. If their sanitation waste was to be monitored, respondents wanted the results of the data analysis communicated back, and especially at the camp. I do want to acknowledge who is left behind when sampling in Malawi or at the camp, because this is better community surveillance, but not full coverage. So what are the boundaries in the humanitarian context? And this applies to Malawi and many low and middle income countries. If I can't send a technician to collect a sample safely at a pit latrine, I won't, such as an unimproved pit latrine that may collapse. There's also sandy soils along Lake Malawi, where pit latrine collapse is frequent, and people with disabilities disproportionately do not have access to safe sanitation facilities. Thank you to the outstanding team at the University of Louisville and many others that make this work possible. Thank you.
Hi, Rochelle. Thank you so much for that Thank you so much for that description of the program. And I'd like to invite all of the panelists and presenters to turn on their video to answer questions now that have come in. And again, I'd highlight for all the attendees, please write your questions into the Q&A. We're both answering them live in the Q&A, and we'll answer some, we're answering some via text, and we'll answer some here as well. So one of the first questions that came up that I wanted to first hand to Marlene and then invite anyone else to answer as well is, what are the main challenges faced in implementing wastewater surveillance systems in humanitarian response contexts, and how can we overcome them to ensure the safety and health of affected populations? So over to Marlene.
Thanks, Danielle. Yeah, that's a very big question. So I'll try to tackle a couple facets of it, and I'm sure everyone has something to say about this one. First, I would say, of course, all of the sort of typical challenges that we encounter in terms of implementing programs in humanitarian contexts are going to apply, right? So that is having personnel, funding, the logistics of setting up a system like this, and security and access concerns in particular. We really heard that in the presentation from Ross today, right? So that's a significant challenge in starting these programs. But another thing, another couple areas to touch on. One is training. That came up in another question. A lot of the training for doing wastewater monitoring and the techniques that are required are similar to techniques that are required for clinical testing. And so there is a lot of overlap in skill sets and a lot of ways that people who are, already have quite a bit of training may be able to cross over into wastewater monitoring space. But training to actually do the testing is one important need, along with the equipment and supplies in order to be able to do the testing. And so that's one thing that has really come up recently. That's a big difference that I think is emerging in some of the work that's happening in high-income countries, and work that is maybe more feasible in humanitarian contexts in particular. Rochelle talked about a lot of the culture-based testing of a lot of these pathogens. I talked about how polio, a lot of the history is based on culturing polio. And a lot of the recent work that we've been doing on detecting this wide array of infectious diseases is based not only on PCR, but on digital PCR, which requires expensive instruments, more training. And I can tell you, because we've spent literally years trying to get some of these systems set up in Bangladesh, in Dhaka, so that we could do work in Cox's Bazar. And it's been a huge challenge to maintain the supply chain to get that digital PCR up and running and make it consistently available, even in labs at ICDDRB, where there are so many highly trained scientists, and they have actually an enormous amount of equipment that they use for projects on a consistent basis, and yet still the digital PCR has been difficult to keep that up and running. So some of those newer cutting edge technologies, we really need to focus on how can we bring those into a format that are more globally accessible, so that we can actually use those tools in humanitarian settings more effectively. And then the final challenge that I'll note is what the sample represents. So one benefit of wastewater and environmental monitoring is that the sample is going to represent whoever is there at a given time. So regardless of whether you have a really good, accounting of the population, you're going to have a representation that does represent that population, with some of the caveats that Rochelle was describing. And so it's a good real-time representation. At the same time, if you have flows that are really inconsistent, if you have samples that you take that are representing relatively small communities, and you have to take many of them and combine them to get the community as a whole, the logistics of that can be challenging. And it's more challenging to get a quantitative result from those samples. It is much more achievable to look for the presence and absence, or maybe strong presence, of a particular target that you're looking at. But that some of the epidemic curves that I showed, where we look at the quantity over time, if we don't know exactly what the inputs are, then it's challenging to control for those in a way that helps us construct that quantitative timeline. So yeah, I will stop there. I've talked enough.
Oh, that was great. Thank you. Do any of the other presenters want to add into that? in terms of challenges and ensuring safety?
Yeah, I think from a cluster perspective, really one of the challenges is trying to make sure that you can get buy-in across the humanitarian community. I mean, if you're in a humanitarian context, when we were first in Gaza, UNDP, the lead on solid waste, wasn't even in country because they had been ruled as not important enough at that particular point in time. And we managed to demonstrate that over time, and therefore a massive response came in. Being able to get across that wastewater testing is a key activity to be able to do to protect the population. It's something you actually have to do. I know speaking to this group that doesn't necessarily make sense, but when you're actually speaking to non-technical people who are deciding about vehicles moving around, it is hugely important. So how you communicate on this and what you're saying, you can't just do technical speak. You have to portray it in the right way. Over.
Yeah. Anything else? If I can add, and to link to another question that was asked on training, I think there's 3 layers of training that need to be completed for this work. And the first is around the sample design, where to collect the sample, how to collect the sample, what might that sample mean if you collect it from a wastewater system or a latrine, who's being left out, as others said, who's being included in that sampling. And that's a sample design training. The second training I would say is on safety of collection. And I think the video that Rochelle showed was fantastic at showing the PPE necessary, the personal protective equipment, the how safely these need to be collected. These are infectious samples. And we can't just send people out in no PPE to collect them, not thinking of their own health and how to keep those samples safe to the laboratory. And then, as Marlene spoke about so eloquently, thinking about actually the training that needs to happen at the laboratory level to ensure the samples are appropriately analyzed, maybe qualitatively or quantitatively or with different methods for different diseases. And so I think this isn't one of the things maybe coming across from all our speakers is this is something that takes a lot of time, right? This is something takes a lot of effort. And sometimes for small samples, Ross, I think you said there were 8 samples in Palestine and there were kind of in the number of tens of samples in Cote d'Ivoire. Malawi seemed to have more samples over time, but this is a more complicated procedure. And so I don't know if anybody wanted to add anything into that.
I can jump in a little bit on training. We calculate we need 17,000 water, sanitation, and hygiene, I guess, practitioners in Malawi. And I would put WBE into the WASH category in Malawi because there's not a dedicated WBE training program. Currently, we are using medical microbiologists for our sample collection and analysis. We're only running culture method because we can't get the supplies reliably for PCR. We can get them about once a quarter realistically. So one of the things that we are doing to expand this is we did do desensitization at the camp prior to collecting the camp samples. And the last 2 years, we've run a Girls Science Day camp, which has included 250 elementary or primary school girls to let them know that water, sanitation, and hygiene, wastewater-based epidemiology, and public health could be careers that they may be interested in joining. And so 500 girls in total in the last 2 years, and we look forward to expanding that. But I think we need higher education programs to train more students before we can really expand this as a public health tool in countries like Malawi.
Thank you so much. And your point echoes points of Marlene and Ross, which is about the long-term training needed to really start these programs. There's one question from Helen or Helene. We didn't talk so much about infectious parasitic diseases. Marlene, do you want to talk a bit? Are parasitic diseases also included in waste effluent surveillance?
This is a great question, and maybe other speakers on the call have more direct experience with this. I think that, a lot of the recent focus for wastewater monitoring has been on viral diseases because those are some of the newly and rapidly emerging threats, and because our systems that we've been developing work really well for detecting and quantifying viruses. But there's a very long history of, I'm sure all of us have worked in various ways who are presenting today on detecting and quantifying parasitic diseases in the environment. And so this can definitely be applied to wastewater monitoring. And I have heard of a couple projects where this has been done. However, I don't think it has been a focus globally as much as some of the viral and then the kind of enteric bacterial diseases.
Yeah, exactly. There's a question from Brian Reed, which is, who are the results for? It sounds like this data is for the health sector, not so much the WASH sector. And I wanted to turn that over maybe to Ross or Alyosha to talk a little bit about how the WASH and health sectors might work together in humanitarian response. And I'm not sure, Ross or Alyosha, which one of you wants to jump in on that question?
Hi, yeah. I mean, I can certainly start on that. It's very much a WASH area. It's also very much a health area. And therein lies a challenge. We need to make sure that health and WASH are working very closely together. Typically, we've had a response which is, oh no, there's a crisis. Oh no, we've got a highly infectious disease. Cholera is spreading. What kind of response is going to happen? And actually, through wastewater sampling, we can get ahead of the curve. And the tool we use to get ahead of this is infection prevention control, planning for it. And the way that works best is if it integrates both WASH and health objectives. So you bring the two groups together, you discuss what are the likely risks, you discuss and decide how the two teams can collaborate, both in terms of prevention and in the event of a response. And this is the best way to be able to save lives and to be ready for an outbreak. But that takes quite a lot of work to bring together, because you really have to create the interest in the other side. And speaking as a WASH person, you have to create the interest within the health actors in order to get them to engage, and make sure that the data that they're creating is useful to WASH. They're very much more focused at what happens at the health facility level and less focused at what happens at the community level. So there's a few dynamics around that. Over.
Absolutely. And I think this comes back to, for our outbreaks, it's been incredibly important for the WASH sector and the health sector to work together, to share data. We talk about this in cholera with understanding where the cases are so that we can provide targeted response. And I think specifically for our infectious disease work, it's important for the WASH sector and health sector to work together. There was a question here from Bernd. Do you see a chance to connect better this type of waste effluent surveillance to the global efforts brought forward under GLOACON initiative? What would it take from your side? And I have to say, and I am not familiar with GLOACON, and I'm not sure if other people are. Or Bernd, if you can, Bernd, if you could type in or unmute and provide more information. I don't know if any of the panelists want to address that one? all of us
I
made.
don't want to speak for Bernd on GlowCon. Hopefully he can type something in or add more, but I just want to say in terms of the connection between humanitarian contexts and our effort towards surveillance in these settings and the other global surveillance efforts that are going on, I think there's a really important, like I said, connection and feedback loop between the efforts that are happening in both communities that needs to be hopefully better connected, because there are a lot of new tools that are being developed, a lot of new pathogens that we're focusing on understanding whether we can monitor. We need to include pathogens that are of importance in humanitarian settings in our development of testing that is happening in high-income country settings. And we also need to work on those technologies that we're using that are more sensitive and continue to develop the technologies to make them more globally accessible and cost effective too. I think that the sustainability of these efforts globally will depend on us demonstrating that we can create an interconnected global system and that we can do so in a way that's consistently cost effective. And so our sort of mutual learning along that path will help us to achieve that in a way that will be globally advantageous.
Yeah, and thank you so much for that detail in the chat about the work with World Health UNEP and Gates on the global consortium. And I think Marlene's answer is exactly these efforts in humanitarian settings should be linked into the larger work as well. And thanks for that link. And I think we have about 5 minutes left, and I want to give each of the presenters a moment to speak before the end. And I think one of the things that's really come out of this webinar for me, listening to all of these, is that there might be a perception that waste effluent or wastewater surveillance is the new thing, and you can just go grab a sample and have it analyzed and get a result. And certainly here in all the programs that were presented, there's been kind of quite a bit of work to be put in to understand where and how to sample, to train the collectors and to train the labs. And so how I would ask the presenters first if they have anything else they'd like to add before we end, but also to think about how this could move forward, and how you see the training needs, because these are complicated programs, moving forward into the future. I'm not sure if that makes sense, but maybe I'll go in order of how the presenters presented. So Marlene, then Ross, then Yapi, then Rochelle. Does that make sense? Over.
Sorry, I'll unmute. I'll speak quickly because I think I've talked a lot, but I just want to say I really appreciate the way that this session has been constructed to include all of these different perspectives, because I think, again, I'm just going to say it again, these connections that we make between how we operate in different settings and what we can learn from how we make this technology accessible is going to be what makes it ubiquitous. And we have, certainly in the United States, I think we're likely to have some upcoming funding challenges and support challenges. And this is one of the systems that we can have as kind of a backbone to be able to say, we know what's going on in the community as a whole, even when we're in challenging contexts and challenging situations where we have, some of our typical clinical surveillance systems are disrupted. That's something that we're dealing with everywhere. And so continuing to make these tools more accessible and cost effective will enable the sustainability of these systems.
Thank you. And then Ross?
I'd just talk about the opportunity behind it. I think the sampling really exposed a massive problem which would have escalated, and we managed to do something around this in basically the most constrained operating environment that I've ever worked in. And that demonstrated massive collaboration across multiple UN organizations, cooperation across that hostile frontier to move materials, And so it is a fantastic opportunity. And the question came, well, what about cholera? Why weren't you testing for that? Well, it's not endemic in the area. Cholera would have to be introduced. But what a fantastic way to be able to screen for it. If we were actually doing this as a routine, screening on a regular basis, we would be ahead of the curve. So it's an amazing opportunity that's being offered. We just need to find out how you can market it in the right way, both within the humanitarian community and amongst ourselves.
Exactly. Thank you for that, Ross. And then Yapi, I'm not sure if you wanted to add something to end. Thank you.
Okay, thank you, Prof. Thank you, Marlene. Thank you, Ross, for your explanation about your activity. So I'm very happy to participate to this webinar. And about Côte d'Ivoire, what I want to say is wastewater surveillance is a very new activity. It's very new. So we try now to work with the National Institute of Public Health to see how to put the wastewater surveillance in the system of in the epidemiology system, how we can do the correlation with the results of the wastewater and that we have in the hospital. Is it the same thing or not? So the work in Côte d'Ivoire is very big about this activity. So I'm very happy to know new things in this webinar, to know that we have the GlobalCon, that is a global consortium for wastewater and environmental surveillance for public health. And I think that will help us to do other things to help the country to move on. So I'm very happy. Thank you very much for this webinar. Thank you.
Thank you. And then in our last minute, Rochelle, do you want to have final thoughts? And then I'll wrap up.
I think the intersection of WASH and wastewater surveillance, it allows us another way to advocate for disadvantaged communities. And so that's what I'm excited about, continuing in Malawi and many other similar contexts.
Yeah. I wanted to say thank you so much to all our presenters who did such an amazing job of presenting the overview of wastewater and waste effluent surveillance and then example programs of how it can be done. Thank you everyone as well for being here. If anyone has any technical questions or follow-up, please feel free to email. And we just wanted to say it was great to hear, and a huge thank you to all our presenters and attendees, and have a nice morning, afternoon, or evening wherever you're at. Thank you.
Thank you. Thank you, Daniela. Thank you, everyone.
Bye.
You.
Thank you.
Bye.